All right, doctor, whenever you're ready.
All right. Good afternoon. Welcome. I expected less people, but there you are. Congratulations. I'm Dr. Mareș Miseli, and today we're going to start a new block, inflammation and repair. Regretfully, I have no conflict of interest to disclose related to materials. Features of the block, briefly. You will have 3 lectures. This is number 1. Tomorrow is going to be the 2nd lecture. Next Tuesday, the 3rd lecture. You have the CPCs, you have the problem-solving exercises, and the assigned readings that you know about. I don't want you to forget about this repetitio est mater studiorum, one of my favorite Latin proverbs, who says repetition is the mother of the study. I have to warn you about this chapter in particular, that it takes a lot of repetitio. It might look arid at the first glance. There's a lot of information. You will have to do some memory, but the good news is it has a lot of logic in it. Okay, today's lecture's objectives are few. Among them, at the end of this lecture, you will know the mechanism and mediators of acute inflammation. You will learn about potential therapeutic strategies based on these considerations, and you will learn the differences between a transudative effusion and an exudative one. I don't know how you learn, but when I start a new subject, before I go into the nitty-gritty, into the minor details, I need to know the map, the floor plan, if you want. So this is the outline of the whole chapter. We will start today with acute inflammation. We will learn principles of acute inflammation, the steps involved in this inflammatory reaction. You will learn about the key participants in this reaction, inflammatory reaction. You will learn about mediators of inflammation. At some point, not in this lecture, but in the next lecture, you will learn to differentiate between the morphologic patterns of acute inflammation. And then when we will study the outcomes of acute inflammation, you will learn about the chronic inflammation and the second step, the repair. Now let's start with the definition of inflammation. How do we characterize inflammation? What is inflammation? Well, to begin with, inflammation is fundamentally a protective response needed for survival. It can be defined as a response of vascularized tissues to injury. And by injury, we understand infections and tissue damage. This response will bring cells and molecules of host defense from the circulation into the sites where they are needed. And the purpose is to eliminate the offending agents. So this inflammatory reaction has the purpose of eliminating this primary injury, this offending agents. How is it doing that? By destroying, diluting, or isolating the agent or the affected tissue. It will also eliminate the consequences of this process and initiate the process of repair. How do we denote the presence, how do we indicate the presence of inflammation in a particular site or tissue? We take the name of the tissue and we add the suffix itis. And I'm sure you all heard about appendicitis. And then you can do this suffix adding to any tissue or any organ, and you get to denote the inflammation, the presence of inflammation in that particular place. Salpingitis, the inflammation of the fallopian tube. Pericarditis, arthritis, cellulitis, to name just a few examples. Now, how do we characterize the inflammatory reaction using the time criterion? But first of all, we classify inflammation into acute or chronic. And let's talk first about the acute inflammation. What is the acute inflammation? It's the immediate reaction of the body to an injury, to an injurious agent. This is a fast reaction. It starts within minutes or hours. It also subsides pretty fast, within hours or days. The main thing that happens, the main event that happens during this acute reaction is the exudation of the fluid and proteins into the tissues and migration of some cellular infiltrate, which is made mainly by neutrophils. This tissue injury in an acute inflammatory reaction is usually mild and is self-limited, but the local and systemic signs can be prominent. As opposed to the acute inflammation, in case of a chronic inflammation, the onset is slow. This is a protracted reaction. The cellular infiltrate is different. You will not see neutrophils in the tissues at the place of the injury, but you will see mostly monocytes or macrophages and lymphocytes. The injury may be severe and progressive, but the local and systemic signs are less. Now, I think I had this sign before in another slide. Do you know what it means? It means that if I light this flashlight in your eyes at 2 o'clock in the morning and I ask you, what is the cellular infiltrate made of in chronic inflammation, you will just wake up and say lymphocytes and monocytes. You have to know that. So whenever you see this flashlight, it means pay attention, pay more attention than usual. All right, let's talk about the cardinal size of inflammation. And this is a bit of history that, I don't know about you, but I like history of medicine. Here's a Roman encyclopedist and possibly a practicing physician called Aulus Cornelius Celsus, to not be confused with Celsius, which is a Swedish astronomer who proposed the Celsius temperature scale. Aulus Cornelius Celsus wrote an encyclopedia, and in that book, he described many things, among them, the 4 cardinal signs of inflammation: heat, pain, redness, and swelling. And I'm going to tell them in Latin: tumor, rubor, calor, dolor. Tumor, swelling, rubor. How are you going to remember that? Ruby, from ruby, redness. Calor and dolor. It helps if you know a bit of Spanish. Calor, heat, dolor, pain. And in the 19th century, Dr. Rudolf Virchow added the 5th cardinal sign of inflammation, functio lesa, which means loss of function. Now, if you want a bit of a drawing to help you memorize that, here it is. Tumor, rubor, calor, dolor, and functio lesa. Listen to my voice. It sounds like a poetry. Tumor, rubor, calor, dolor. Easy to remember, right? Right. All right. Now let's talk about causes of inflammation, some general features of inflammation. Among the causes of inflammation, of course, on top there are infections, but these are not the only causes of inflammation. Tissue necrosis by different mechanisms, by ischemia, by mechanical factors, trauma, by physical or chemical injury, extremes of temperature, and so on. Tissue necrosis can also incite an inflammatory reaction. Foreign bodies. And also, don't forget about immune reactions, which can persist with features of chronic inflammation. Now, these are the causes. The inflammatory reaction will be induced by chemical mediators. Very, very important point, and one that's gonna give you some trouble, I suspect, because you will need to memorize the names and some of their actions. So the chemical mediators will be produced by host cells in response to injurious stimuli. The outcome of acute inflammation, we will spend some time for this, but right now just remember that either it goes or it stays. So it leads to elimination of the noxious stimuli, followed by a decline of the reaction with a repair of the damaged tissue, or there is a persistent injury resulting in chronic inflammation. I don't know if you heard about my lectures, but this is one particular that I have. I'm known for the Zen moments. So once in a while, I'm just going to flip you out of your concentration, and I'm going to try to change your perspective of the world. And I'm going to give you the so-called Zen moment, where I pick something that's funny for me. I hope it's funny for you. So here's the first Zen moment. In order to keep a good perspective of one's importance, everyone should have a dog that admires him and a cat that ignores him. I have 2 cats. All right, let's go back and let's take a look at the sequential steps in inflammation. Now, look at the picture. These are the offending agents. The offending agents located in extravascular tissues will be recognized by host cells, by macrophages, by dendritic cells, by mast cells. So this is the first step, recognition, R, okay? in turn, these cells will synthesize the mediators. The mediators will act upon vessels, and they have two consequences. They induce vascular reaction and a cellular reaction. Now, in terms of vascular reaction, they will lead to vasodilation and increased vascular permeability, which in turn lets fluid leave the vessel and go into the tissue, and the clinical consequence of this mechanism is edema. Now, leukocytes and plasma proteins will be recruited from the circulation to the site where the offending agent is located. So this is the recruitment. The next step is their activation, and then they will work together to destroy and eliminate the offending substance. For that, they have to leave the vessel and go into the tissue. Here, they work towards the removal of the offending substance. And the whole reaction will have to stop at some point. Otherwise, it's going to be more destructive than the injurious agent. So the reaction is controlled and is terminated. So this is called the regulation of the reaction. And the last step is repair, where some other types of mediators will act upon fibroblasts and extracellular matrix, leading to the initiation of the process of the repair, which is a subject on its own for the last lecture. Now, I mentioned vascular reaction and cellular reaction, and this is another picture that shows you more detail about these said chemical mediators and vascular reaction and cellular reaction. And if you look here at this picture, you'll see a mast cell, which is a source of mediators, a macrophage, which is also a source of mediators. And there's another source of mediators. If you look here, this is the endothelium. I hope you remember the structure of a blood vessel. This is the inner lining, the endothelium, basement membrane, and the smooth muscle. And do notice the space between the endothelial cells, which is normal here. It's pretty tight. And then there's a gap. There's a bigger space between the endothelial cells. So that's a vascular reaction. The vessel will undergo vasodilation. They are also a source of mediators. On the other hand, the cellular reaction involves the polys, the polymorphonuclear leukocytes, which will pass through these gaps between the endothelial cells and go to the tissue, where they lead to elimination of microbes in that tissue. Other participants to this cellular reaction are the plasma proteins, and among them complement and clotting factors. Lymphocytes also participate, and also monocytes leave the vessel and go into the tissue, and they become the macrophages. And not only they are the source of mediators and they have a role in the immune response, but they also help with the elimination of microbes and that tissue. And here in this schematic shows you the process of repair, which takes place afterwards. Now, the first step in inflammation is the recognition of microbes and damaged cells. How are the microbes recognized? By cellular receptors. So there are cellular receptors for microbes expressed by many cells, most importantly by phagocytes and dendritic cells. The most important class of these cellular receptors are the so-called Toll-like receptors, TLRs, named by the founding member Toll, which is a gene discovered in Drosophila. These receptors are located in the plasma membrane, but also in the endosomes. They will detect extracellular and ingested microbes, and they also recognize pathogen-associated molecular patterns. The consequence of activation of these receptors are few. Among them, the most important are stimulation of the secretion of inflammatory mediators. They increase the secretion of antiviral cytokines, the secretion of lymphocyte activation proteins. They also increase expression of adhesion molecules on the cell surface. Are you bearing with me? I told you it is a bit arid. I tried to make it as colorful as possible, but this is what it is. This is basic science. So you have to memorize that part. Logic comes though. Another method of recognition is by sensors of cell damage. All the cells, aside from those receptors, they have cytosolic receptors that recognize molecules altered as a consequence of cell damage. And they activate a so-called inflammasome, a multi-protein cytosolic complex, which in turn will induce inflammation and produce the cytokine interleukin-1, which is also a mediator of leukocyte recruitment. What is the clinical relevance? Well, you can think about many instances where there are substances that accumulate in the cells or outside the cells, they damage the cells and they activate this inflammasome. For instance, in gout, where you have accumulation of the uric acid crystals, or the atherosclerotic plaques, where you see cholesterol crystals and so on. Another way of recognition of microbes or damaged cells is by circulating proteins. Most important among these proteins is the complement system, which you know reacts against microbes and produces mediators of inflammation. Another important class of circulating proteins are the mannose binding lectin, which will recognize microbial sugars and promotes ingestion of microbes and activation of the complement system. And finally, collectins, which bind to microbes and promote their phagocytosis. Now, fun starts. Mediators of inflammation. How are we going to define the mediators of inflammation? These are substances that initiate and regulate inflammatory reactions. And here on the left side, I just enumerated the most important classes of mediators: the vasoactive amines, the lipid products, prostaglandins and leukotrienes, cytokines, including chemokines, and the products of the complement system activation. On the right side, I'm not going to go through all that table. That's for your study later on. But I want to draw your attention upon, to some points. So you see here, they start with histamine, the vasoactive amine, most important one being histamine, prostaglandins and leukotrienes. These are the lipid products. And then cytokines and chemokines and so on, complement and kinins. These are the main sources of these mediators. Do not try to memorize that at the first reading. Just keep coming back to this table. Read, come back. You will forget. It's impossible to memorize everything from the first reading, but just repeat, come back to this table. I want to draw your attention upon two things. First is repetition. If you look here, you see vasodilation, increased vascular permeability, vasodilation, and so on. Things repeat. They're pretty similar in their effects. But I also want to draw your attention to some other action, the systemic action, among which most important are pain and fever. Systemic effects, fever, systemic effects, pain. So keep in mind that these mediators can have systemic effects, most important among them pain and fever. If you want to look at the problem from the other angle, from the opposite angle, you can learn what are the main reactions of inflammation, such as vasodilation, increased vascular permeability, fever, pain, and so on. And you can look at this table and see which are the principal mediators who have this reaction. Whatever way you want to learn it, just keep coming back to these two tables. Now, how do we classify the mediators of inflammation? We classify them into plasma-derived mediators or cellular-derived mediators. And look at the first part of the drawing here. The plasma-derived mediators usually are, you find them in the plasma, obviously, as inactive mediators. They will need to undergo a series of cleavages in order to achieve their active state. Most of them are produced by the liver, and if you look at the source, you will see, you already know this system, the kinin system, the coagulation system, and the complement system. They are active against circulating microbes, and if there is a need, they can be recruited into the tissues. The cellular-derived mediators, they can exist either preformed in secretory granules, or they can be newly synthesized. Here you have the most important ones, the examples of the most important ones, and keep in mind that they are produced mainly by macrophages, dendritic cells, and mast cells, but all leukocytes, neutrophils mainly, platelets, endothelial cells, and most epithelial can be induced to produce these mediators. Now let's take a quick look at each of these big classes that I mentioned. The first class is the vasoactive amines. First, most important of them, histamine and serotonin. These are the two major representatives of this class. Histamine, the main effect, I list here the main effects. Histamine leads to dilation of arterioles and increases the permeability of the venules, while serotonin is a vasoconstrictor. They are stored as preformed molecules in different types of cells, most importantly in mast cells, in basophil, and in platelets, and they are released in response to many factors. Most importantly, physical injuries such as trauma, cold, or heat. Binding of antibodies to mast cell also can lead to release of these vasoactive amines, and you see that in allergic reaction. They also are released as a consequence of the action of anaphylatoxins, and also in response to neuropeptides and some cytokines such as interleukin 1 and 8. The next class of chemical mediators are the so-called eicosanoids, or the lipid mediators. I don't know if you remember from biochemistry, eicosa is a prefix used in chemistry to indicate 20. Why? Because arachidonic acid is a 20-carbon polyunsaturated fatty acid. What happens in this case is the different mechanical, chemical, and physical stimuli or other mediators will activate the phospholipase A2, which is an enzyme which acts on membrane phospholipids and converts them into arachidonic acid. In turn, arachidonic acid will be converted either into prostaglandins under the action of these enzymes, cyclooxygenases, COX-1 and COX-2, or under the action of lipooxygenases will be converted into leukotrienes and lipoxins. So these two are called lipid mediators, these two classes. Now, this is a drawing which I like for a few reasons. It gives you more detail. As I said, do not try to memorize all of these details from the first reading. Just try to understand. So it basically tells you the same thing that I said before. The cell membrane phospholipids under the action of phospholipase will be converted into arachidonic acid. And then on this arm of the reaction, you see cyclooxygenase, which leads to formation of prostaglandins and prostacyclins. And here you have some of their effects. And then on this arm of the reaction, and you see that under the action of the lipooxygenases, arachidonic acid is converted into leukotrienes and lipoxin A. What is also important here is some very, very practical consequence of knowing this reaction. You see here the antagonists of this reaction. So steroids will stop this reaction. COX-1 and COX-2 inhibitors will stop this reaction, though, will stop the formation of the prostaglandins. And also lipooxygenase inhibitors will stop this reaction. And notice this one, leukotriene receptor antagonists will prevent all the leukotrienes by from acting on the tissues. So they have one very practical application. In clinic, they used to treat asthma. They prevent the bronchospasm induced by leukotrienes. Cytokines and chemokines are another class of chemical mediators. Cytokines are proteins secreted by many cell types, mainly by activated lymphocytes, by macrophages and dendritic cells, but also, as I mentioned before, endothelial, epithelial, and connective tissue cells. These cytokines will mediate and regulate immune and inflammatory reactions. A family of small proteins that act primarily as chemoattractants for specific types of leukocytes are called chemokines. In this table, you see a few examples of the main cytokines classified into acute inflammation and chronic inflammation, classified by the type of reaction where you see them mainly. But I want to draw your attention here. It says there's also considerable overlap between the cytokines involved in acute and in chronic inflammation. And if you see here, interleukin 17, just to give an example, it's involved in acute inflammation as well as in chronic inflammation. And here you are given the principal sources and the actions in inflammation. Again, do not try to memorize all of it. Anybody knows who this guy is? Do you guys like to read? I mean, aside from medical books? Yes? No? Anybody? Oscar Wilde. You do? You recognize him? No? That's Oscar Wilde, one of my favorite authors. He said, very, very smart guy. He said, it is a very sad thing that nowadays there is so little useless information. No? You don't like it? I think it's right. All right, so now let's take a look at the steps of the acute inflammatory process. And let's start with the reactions of the blood vessels in acute inflammation. When you talk about the reactions of the blood vessels in acute inflammatory reaction, you have to think of three major components. One, dilation of the small vessels, which leads to an increase in blood flow. Two, increased permeability of the microvasculature, enabling the plasma proteins and leukocytes to leave the circulation. And three, the third step, migration of the leukocytes from the microcirculation, their accumulation in the focus of injury, and their activation to eliminate the offending agent. You have to admit that there's a lot of things to memorize, but there's a lot of logic. I think these steps are very, very logical. So here's how my logic works. When I think about vasodilation, this is what I came up with to memorize easier. Vasodilation, it begins early. You have to remember that, and may be preceded by the transient vasoconstriction, few seconds. It is induced by few mediators acting on smooth muscle, and it involves first the arterioles and then the capillaries. Vasodilation, in turn, will lead to an increased blood flow. How does this translate clinically into heat and redness, which is something that we call clinically erythema? When you see some red area, you call it erythematous area, erythema. Not only there's an increased blood flow, but there's an increased permeability. I showed you the spaces between the endothelial cells. They become larger. This in turn leads to loss of intravascular fluid. Where does it go? To the tissue. The process is called exudation. Exudation, remember, you're going to remember this word from Exodus. Exudation, just leaving the vessel, going into the tissue. What is it left? What's left behind? A slower blood flow with an increased viscosity. It's a thicker blood and slower. We call this stasis, a stagnant blood, stasis. Histologically, we call it congestion. Clinically, it translates into erythema. There's a consequence to this stasis. The leukocytes will undergo the process of margination, which just means they will go to the margin. They would go to the sides of the vessel. So they will accumulate along the endothelium. The activated endothelium will express adhesion molecules, and the leukocytes will roll, will adhere, and then they will migrate to the vascular wall. We're going to come back to this. I have a whole slide showing you this process. Now, I want to introduce new terms, new vocabulary. I already said exudation. Exudation is the process of escaping from the blood vessel, escape of fluid, proteins, and blood cells from the vascular system into the interstitial tissues or body cavities. We call edema or effusion the excess of fluid in the interstitial tissue, that's edema, or serous cavities, that's effusion. This fluid can either be an exudate or a transudate. Am I just going to know by looking at it grossly? Maybe, but what do I need in order to classify this fluid into an exudate or a transudate is some lab, some chemical tests. So how do I define an exudate? Exudate is an extravascular fluid that has a high protein concentration and contains cellular debris. Exudation is due, the exudate is due to increased impermeability of the small blood vessel, typically during an inflammatory reaction. Now, I mentioned here a special type of exudate. It's called pus. You all know pus. This is a purulent exudate. It's an inflammatory exudate rich in leukocytes, mostly polys, debris, and microbes. This is just the component of pus. On the other hand, transudate is a fluid, extravascular fluid, with a low protein content. You notice the differences right now? Exudate, high protein content, transudate, low protein content, little or no cellular material, and low specific gravity. This transudate is a result of osmotic or hydrostatic imbalance across vessels with normal vascular permeability. So let me repeat, let me give you the keywords. Exudate, high protein, inflammation. Transudate, low protein, normal vascular permeability. Now let's take a look to the actual mechanism. And let's compare them again, exudate versus transudate. The first drawing shows you the normal vessel with the normal pressures. Hydrostatic pressure, if you remember from physics, the hydrostatic pressure is the pressure exerted by a column of fluid on the walls of the container. That's the hydrostatic pressure pushing out. Colloid pressure is exerted by the plasma proteins, is the pressure that keeps the fluid in the vessel. So you have to have a balance between these two pressures, hydrostatic and colloid. In the case of an exudate, notice here, most important word that I want you to notice is inflammation. Why? Why do you have here? Because you have, as a result of inflammation, you will have increased interendothelial spaces, you have vasodilation and stasis, and you have fluid and protein leakage. So the result of this inflammatory process will be exudate. Fluid outside the vessel, high protein content, may contain some white and red cells. On the other hand, a transudate will have low protein content and few cells, and an exudate will be due to an imbalance in these two forces. Either you have too high of a hydrostatic pressure, and you can think of different instances where you can see that, for instance, congestive heart failure. You have an increased hydrostatic pressure, or an instance where you have decreased colloid osmotic pressure. Either you push too much on the vessel wall, or there's nothing to hold the fluid inside. And for that, you can think of instances such as liver disease, where you don't have enough protein synthesis, or you have enough protein, but you lose it through a kidney disease. So these are two instances where you have decreased colloid osmotic pressure, which can lead, either one of these situations can lead to the formation of a transudate. You got it so far? Okay, good. And if you didn't get it, here's more detail. I'm not gonna repeat it, okay? Comparison between exudate and transudate. I put it in red here. Protein content, more than 2.9 grams per deciliter. It coagulates. Why? Because it contains fibrinogen, which is a large protein. Large protein, inflammation, big spaces will leak into the tissue. As opposed to that, no fibrinogen, only albumin, which is a small protein, no coagulation when we're talking about the transudate. And then the differences continue between the two of them. Try to memorize this as much as you can. Not all the numbers, but just know the main differences between exudate and transudate. It's important. Now, I mentioned increased vascular permeability, and I need to tell you there are few mechanisms by which this vascular leakage can appear. Most important of them, most common of them, is the endothelial cell retraction. I showed you before this in large spaces. It's the widening of the intercellular gap of postcapillary venules. It's early. You see this 15 to 30 minutes after injurious agent. Excuse me. It occurs rapidly after binding of histamine, bradykinin, or leukotrienes, or it can be slower. It can take 4 to 6 hours to develop, and it can be more prolonged after the action of some particular cytokines, such as interleukin-1 and tumor necrosis factor. Another mechanism by which we can see increased vascular permeability is direct endothelial cell damage, resulting in necrosis or detachment of these endothelial cells. You see this in severe injury, such as chemicals or burns or toxins. It affects all the vessels, venules, capillaries, arterioles, all of them without discrimination. Damage of the vessel wall by adherent leukocytes can be added to this severe initial injury. This begins immediately after injury. It lasts several hours or days until the damaged vessels are either thrombosed or repaired. And the last mechanism is transcytosis. The contribution of this mechanism to the vascular permeability is unclear, but we mention it here. It means increased transport of fluids and proteins through the endothelial cells. It takes place mainly in venules. It may involve intracellular channels that open in response to certain factors such as VEGF, vascular endothelial growth factor, that promote vascular leakage. How about the lymphatic vessels? We just spoke about the blood vessels. What about the lymphatic vessels and the lymph nodes? Do they participate in an acute inflammatory reaction? Of course they do. Lymph flow will be increased because the lymphatics will drain edema fluid. Also, the lymphatics proliferate during inflammatory reaction for the same purpose, to handle the increased load. Then also you have to keep in mind that they may become secondarily inflamed, and in that case we call it itis, lymph lymphangitis. Lymph from lymphatic, ang from the vessel, and itis from inflammation, lymphangitis. Also, you can see inflammation in the draining lymph nodes, lymphadenitis. And clinically, this translates into red streaks near a skin wound. Are you lost? You follow? How is it so far? We're good? We're good? Can you keep it for the end? Because I don't know how I'm doing with time. But don't forget your question. Write it down. All right, so you're not lost. You're right here. All right. Let's continue with, we just spoke about reactions of the blood vessels in acute inflammation. Now let's take a look at the leukocyte recruitment to sites of inflammation. The leukocytes will be recruited to the site of inflammation, and they will have a key function. They are needed there. They needed to eliminate the offending agent. Here you have another table that compares the polys and the macrophages, which are, I already told you, these are the main components, main cellular components that leave the vessel and go into the tissue at the site of the injury. And they are compared by their origin and lifespan and responses to activating stimuli. Again, this is for you to take some time and study it at your leisure. How do they go to the site of inflammation, to the tissue? By extravasation, delivery of the cells from the vessel lumen into the interstitium. Into the lumen, there are a few processes that we're going to take a quick look, starting with margination, rolling, and adhesion. I already mentioned margination. Then they migrate across the endothelium, mailing the venules. This process is called diapedesis. And then while they get when they get to the tissue, they will migrate in the interstitium by a chemical gradient. They will go towards the site of injury. And this process is called chemotaxis, the chemical gradient, taxis, movement, chemotaxis. So let's take a look at this process. You have to admit, this Robbins book gives you some really nice pictures, right? Right. Okay, so let's talk about the leukocyte migration through the blood vessels. There are a few steps. The first is rolling. This is a low affinity with a fast rate interaction mediated by selectins. And you have here an example of selectin. The next step is firm adhesion. So they roll, they roll, they roll, they go towards the endothelium. Here, there's firm adhesion taking place, mediated by integrins, which are class of leukocyte surface proteins. These are two-chain glycoproteins, integrins, selectins, integrins, rolling, adhesion. The next step is transmigration or diapedesis across the endothelium. You have to remember this, this is important. CD31, this is the mediator. This process, this step takes place due to the existence of the piercing of the basement membrane, probably by collagenases. And then while they leave the vessel, they go into the interstitium and they will move towards chemoattractants. They go toward the place of the injury. And as I mentioned before, they are attracted by these substances called chemoattractants, and the process is called chemotaxis, movement in the tissue along a chemical gradient. Among the most important chemotactic factors are bacterial products, cytokines, some components of a complement system, particularly C5a, and also leukotriene B4. Very important chemotactic factors. Now, the nature of the leukocyte infiltrate varies with the age of inflammatory response and the type of stimulus. This has a very, very important application, very important for pathologists, but not only for pathologists. Now, I want you to look at this graph here and notice that in the first day, edema will reach a peak. And then neutrophils will predominate during the first 6 to 24 hours. There are a few reasons for that. They are most numerous in the blood. They respond rapidly to cytokines, and they have a shorter life in tissues than monocytes. And if you notice here, at one day, they have a peak, and then they start decreasing. And then meanwhile, the macrophage infiltrate, monocyte macrophage infiltrate, will reach its peak after the second day. So over 24 to 48 hours, they will be gradually replaced by macrophages. Let's look in the tissue. What happens here? This is a dilated vessel. This is a section from myocardium after my ischemic necrosis, after an infarction. Here you see, I'm going on the outline of a congested vessel. How do I know it's a vessel? I see a lot of red blood cells here. And what do I see predominating here? Do you remember the cells? You need to know the normal. Before you learn any pathology, you need to know before the histology. So I will show you some pictures of the normal cells. But for now, I hope you recognize this is the polymorphonuclear neutrophil. These cells have 3 to 5 lobes. If you look at this nucleus, you'll see there's not a circle. It has little lobes. So that's a poly. They predominate here. And later on, you don't see these you don't see these polymorphonuclear cells. What you see are these mononuclear cells, lymphocytes or macrophages. In this picture, I see a predominance of the lymphocytes. That is the chronic phase. Let's talk about circulating cells. I asked you if you remember the cells, and I don't want to say I'm assuming you're not, but I just want to make your remembering the cells easier. So this is the look of a neutrophil. This is an eosinophil, bright, red, bright, small granules. Very, very easy to spot under the microscope. Here you see a basophil, big, chunky, very blue, very dark blue granules. This is a lymphocyte, round nucleus, very little tiny cytoplasm around it, dark blue round nucleus. And this is a monocyte, the biggest of all, pale blue cytoplasm. Sometimes you see vacuoles in the cytoplasm, and this big kidney-like nucleus, if you want. These are erythrocytes, and these are the platelets. I think you saw this before. I don't know, I'm not going to ask you to know all these intermediary stages, but I want you to have a clear picture in your head about these lineages. The red blood cells, the white cells, these are divided into the granulocytes and agranulocytes. Granulocytes are basophil, eosinophil, neutrophil, and lymphocytes and monocytes. And these are the platelets. Because I mentioned lymphocytes, monocytes, polys, and I don't remember I don't know if you remember them and how they look like. And also an important number, some important numbers that you should know, 60 and 30, these are percentages. 60% of all the blood cells will be polys, neutrophils, 30 will be lymphocytes. Mono and eosinophils, 6 and 3. You see the same number? 60, 30, 6, 3. And basophils are 1%. Important numbers to remember. All right. Who's this guy? He said, there are only 2 tragedies in life. One is not getting what one wants, and the other one is getting it. How true. You don't know that yet. Okay, good for you. All right, so let's move on to phagocytosis and clearance of the offending agent. So what happens with the leukocytes after they left the vessels? After recruitment, in order to perform their functions, leukocytes, they must be activated by different, by microbes, by products of necrotic cells, by chemical mediators. Why is this necessary? Because activation will enhance the phagocytosis and the destruction of the microbes and the dead cells. It will enhance the release of substances that destroy extracellular microbes and dead tissue, and it will help into recruitment and activation of more leukocytes. But you have to keep in mind that as in any war, as in any battle, there will be some collateral damage. So leukocytes are important mediators of direct injury to normal cells and tissue. This is a part of normal defense mechanism against infectious agents, and you will see tissues at or near site of infection suffering collateral damage. And this is important to know because sometimes some host responses may cause more damage than the pathogen. And here are some examples: autoimmune diseases, allergic diseases, TB, some viral infection, and you probably heard about the COVID-19 cytokine storm. So that's another example of this collateral damage, if you want. Now, other functional responses of activated leukocytes, we already mentioned that they have a role in eliminating microbes and dead cells and in producing cytokines, but they also produce growth factors, which stimulate the proliferation of endothelial cells and fibroblasts and the synthesis of collagen. And they also produce enzymes that remodel connective tissue. This is important because the first two actions are involved in the actual acute inflammation, and these two roles that I mentioned last are involved in the second part, the repair part. So also keep in mind that T lymphocytes, especially T helper Th17, produce which produce the cytokine interleukin 17, contribute to acute inflammation by inducing the recruitment of other leukocytes. Now let's talk about outcomes of the acute inflammation. Well, in a nutshell, it stays or it goes. So it can, after an acute inflammation, the reaction can subside, the injurious agent was defeated, and you can have complete resolution. Or it can persist, and it can evolve into a chronic inflammation. You have to keep in mind that chronic inflammation may follow the acute process or can from the start be a slow, insidious process without this loud stage of an acute inflammation. Also, there are two other possible outcomes of an acute inflammation: abscess formation and fibrosis. In abscess formation, you will see a lot of pus with a lot of destruction of the tissue, and fibrosis usually follows after substantial tissue destruction. Usually you see this in tissues that do not regenerate, and usually after abundant fibrin exudation. All right, so let's take a look here. Same thing that I just mentioned before. Start here. Acute inflammation, different injury. You will see all the vascular changes, mediators, neutrophil recruitment. What can happen? It can evolve into complete resolution, where there's clearance of injurious stimuli, clearance of mediators, acute inflammatory cells. It's like it's never been there. Replacement of injured cells, normal function. Perfect, ideal situation. Or if there's an extensive process, an extensive injury, you can have accumulation of pus formation and accumulation of pus with formation of an abscess. These, when you have so much destruction, the healing usually happens with a lot of fibrosis and some loss of function. And these here, I mentioned two complications of the abscesses formation, ulcer and fistula. Another way of evolution of this inflammatory process is direct healing without formation of the abscess, healing, but if there was a lot of destruction, again, you will see a lot of fibrosis and a loss of function. Now, let's start here, chronic inflammation. Either there's an acute inflammatory process that it persists, and it has a protracted evolution and transforms into a chronic inflammation, or from the beginning you have an injury that evolves directly into a chronic inflammatory process. Usually when you see a chronic inflammation, you will see healing with fibrosis and some loss of function. And now let's talk about termination of the acute inflammatory response. It declines happily. It can, and I mentioned before, this is an essential, it's a fundamentally protective reaction. It's essential for survival, but it has to stop because it has potential to destroy the host as much as the injury. So it declines after the offending agents are removed. Why? There are a few reasons. The mediators of inflammations are produced in rapid bursts. The mediators of inflammation exist as long as the stimulus persists. They have short half-lives and they are degraded after they are released. Those are the mediators. Also, neutrophils have short half-lives in tissues, hours or maximum 1 or 2 days. And the inflammatory process in itself triggers a variety of stop signals that actively terminate the reaction. It switches to anti-inflammatory cytokines. It's a yin and yang. And you might think that this is all very academic and arid, and why are you learning that? But it's not quite so, because the molecular understanding of leukocyte recruitment and migration has provided, all this lab work provided a large number of potential therapeutic targets for controlling harmful inflammation. All this lab work that detected all the steps and all the mediators and all the other substances involved in this acute or chronic inflammatory reaction detected that there are steps where we can intervene. And for instance, agents that block the tumor necrosis factor, which is one of the major cytokines in leukocyte recruitment, it's very successfully used for controlling chronic inflammatory disease. Also, there are antagonists of leukocyte integrins approved for inflammatory diseases. So there are there's a very practical finality of knowing this, all this arid basic science information that I gave you so far. And here's the last Zen moment. You know this guy, right? Woody Allen. He said, confidence is what you do know Woody Allen, right? Okay, good. So you're watching movies, good. Confidence is what you have before you understand the problem. I do hope you did not lose your confidence. This is not such a complicated problem, but I'm gonna stop here. For today. Thank you so much. And we're gonna start tomorrow with the second part. Dr. Lento, how do I stop the video? Stop share. Yes.